Hello students, welcome to the theory class of Principles of Plant Breeding. Today we'll discuss about the emasculation and pollination technique in important horticulture crop plants. So to start with, let's discuss the selfing and crossing program, which are essential part, which are most important part of a plant breeding program, which is production of a hybrid variety. Okay, so in case of hybrid productions, selfing is important for preparation of a parental line. Okay, we cannot directly make a line as a parent, we have to make it homozygous, or you can say true to type, which is possible with the help of selfing, and the crossing program, which is attainable by emasculation and pollination. Okay, so emasculation, what is emasculation and pollination? It is a usual method. It is a mostly flower in self-pollinated crop plants, or in some cross-pollinated crop plants, they are usually bisexual flower. In bisexual flower, we have both androecium and gynoecium. Androecium is the male whole of the flower, and gynoecium is the female whole of the flower. Okay, if a flower is having both androecium and gynoecium, it is called this perfect flower or complete flower. If a flower is having only androecium, it will be called a staminate. If the flower is having uh, gynoecium, only gynoecium, it will be called as pistillate. Okay, so our intention in emasculation as in plant breeding program is to make the bisexual flower into a pistillate flower by removing the androecium. Okay, I think this is clear. And this selfing and crossing program are quite essential in hybridization program. Let's see in the step-by-step procedure of hybridization. First of all, we have to select the parents in case of hybridization program. So after selecting the parents, we have to make inbred lines. By how are we going to make inbred lines? The initially selected parent lines are usually not homozygous. Okay, we have to make homozygous by continuous selfing for 1 or 2 generations, or further generations if it is necessary. Okay, or you can see the example of inbred line preparation in case of hybrid variety lines, hybrid crosses, or in case of pure lines, in case of self-pollinated crop lines, pure line selection is the method. Okay, and right after selection of parents which are ready for right after selfing, the parents are ready for uh, to act as a parent. Okay, and then the emasculation, which is the removal of androecium from the male from the bisexual flower, making it as pistillate flower. Okay, so once we have a pistillate flower, okay, so there is no chances of self-fertilizations. Okay, so now we can introduce foreign pollen into the desired foreign pollen into the emasculated pistillate flower, so that desired process can be attained. So this is our objective. Okay, so two other things are there which are bagging. Okay, what is bagging? It is protection. It is a protection against the foreign pollen. Okay, so in this case, we are the emasculated flower or the pistillate flower is going to be covered with a plastic or butter paper bag, so that it is protected against the foreign pollen before the pollination is done. Okay, as for the rule, emasculation is followed by pollination in every crossing program. So in case of bagging, it should be done in two-step process. One is right after emasculation, okay, and one is right after pollination. Okay, so in case of right after bagging, we need to do tagging. Tagging is quite important. Why? Because right after you bag the emasculated flower, or right after you bag the pollinated flower, okay, in this two-step process, after emasculation and after pollination, if you're going to bag with the same colored bag, or it cannot be distinguished from one another. Okay, which one is crossed, which one is pollinated. So in order to make it distinguished, we can tie a red thread which can be indicative of the cross, or we can indicate with a paper piece of paper, okay, or cardboard paper, where the name of the cross, name of the parent is indicated, and the date of crossing, date of emasculation is indicated, so that it is quite proper in hybridization program. Okay, so let's enter and let's see what are the emasculation and pollination program. Okay, what is emasculation? First of all, it is the removal of stamen or anthers, or you can say androeciums, or killing of pollen of a flower without the female reproductive organ. So we have to retain the female reproductive program, which is reproductive organ, which is gynoecium, and kill, or you can say remove the androecium, okay, which converts a bisexual flower or hermaphrodite flower into a pistillate flower. Okay, so in case usually in case of bisexual flowers, emasculation is essential to prevent the self pollination. Okay, so most of the flowers which comes with both androecium and gynoecium, they are mechanized naturally to allow self pollination in them. Okay, unless and until there is a barrier inside it. Okay, and in case of monoecious plant, male flower is removed. Monoecious plant means plants having either male all male flowers or all pistillate flowers. Okay, so male flowers are removed, and male inflorescence is removed in case of maize. Okay, in case of maize, what happens is both androecium, both male flower is present on the top, which is called as tassel, and female flower is present on the bottom, which is called as cob, later on called as cob. Okay, and in this process, if we have to if we have to make a cross, okay, if we have to make a cross, we can either bag the tassel, okay, so that the falling pollen grains from the tassel, it is protected by the bag. Okay, or it can be removed, the tassel can be removed. Okay, while in case of crop plants where papaya and all, where there is male flowers on some crop plants, unwanted male flowers are to be removed from the flowers, and in case of castor and coconut, what happens is both male and female flowers are present on the same inflorescence, or you can say in the same crop. Okay, so you can remove the male flowers, and you can retain the female flowers. Okay, staminate and pistillate is what you call the male flower and female flower respectively. Okay, in species with large flowers, okay, so emasculation is usually recommended in species with large flowers such as brinjal, okay, and solanaceous crops, okay, where you can see the flower with your naked eye. Okay, and coming to the next method, which is the different types of method of emasculation. Okay, first of all, we have hand emasculation. Next method is suction method, hot water treatment method, alcohol treatment method, cold treatment method, genetic emasculation method, and finally use of gametophytes. Let's see one by one. The most important one and most commonly used one is hand emasculation. In these large flower species which can produce large flower are usually recommended to go for this hand emasculation. Okay, it involves removal of anthers possibly with the help of forceps. Okay, so forceps is the only equipment. Okay, so hand emasculation is usually operated during 4 to 6 p.m. one day before the anthesis starts. Okay, so in this process, it is always desirable to remove other young flowers located close to the emasculated flower to avoid confusions. Okay, if it is not emasculated, so young buds are to be removed so that there is no confusion between the emasculated and unemasculated flowers. Okay, and usually the emasculation is done by removal of only the androecium or the stamens. Okay, or you can directly remove with the along with the corolla and some part of the calyx also. Okay, the corolla of the selected flower is opened with the help of forceps, and the anthers are carefully removed with the help of forceps. Okay, so next method is suction method. It is done with the use of thin rubber or a glass tube attached to the suction hose and is used to suck the anthers from the flowers. Okay, so in which crop plants, it is usually recommended. It is recommended to those crop plants where the flower size is too small, okay, and there is numerous flowers, okay. So example can be rice, or in case of lentil, okay, or in some crop plants such as uh, onions, okay. And hot water treatment, okay, with the pollen grains are more sensitive to female than the female reproductive organs to both genetic and environmental factors. So in this case, if generally in general case, for if we treat hot water, warm water 42 to 48 degrees Celsius for 10 minutes, okay, if we dip the flower, so it can, the pollen can get degenerated, okay. Although this may, the specific amount, the temperature of this hot water treatment may differ from one species to another, so it is a general reference to most of the crop plants, okay. And next is alcohol treatment. So it is not commonly used because of the danger in which it will also in fact affect the gynoecium or the pistillate flower, okay. Usually the generally used dose is 57% of alcohol for 10 seconds is highly effective, okay, and is usually better method of emasculation than suction method. Because why? Because it is, in suction method most of the flowers remain unemasculated, okay, because it is not accurate. And next thing is cold treatment, okay. Cold treatment is like hot water treatment, it kills the pollen grains without damaging the gynoecium. Next is genetic emasculation with the use of cytoplasmic male sterility, okay, and genetic male sterility, so mechanism by transferring the cytoplasmic gene from one wild source to, or related source, to the cultivated ones which you want to use it as a female parent, okay. And next thing is also protogyny and protogyny and protoandry are two mechanisms of differences in their flowering, okay. So in protogyny, the female flower will mature first and the male flower will mature late, okay. So Bajra is a good example of protogyny. So in this case, without emasculation, you can go for we can go for pollination, okay. It is an easy process in Bajra, where the natural mode of emasculation is already there, okay. So use of this is called as natural time barrier in crossing program, okay. So use of gametocides. So gametocides, chemicals such as uh, ethyl sodium methyl arsenate, zinc methyl arsenate in rice, malic hydrazide, such and such are used to kill male gametes without affecting the female gametes, okay. Spraying of uh, right amount of dose of these chemicals can be effective for production of emasculated flowers, okay. Coming to the next thing, okay, bagging, which I have already explained the importance of bagging, okay. It is done right after emasculation and right after pollination also. The importance is it protects, bagging protects the foreign pollen to interfere, to get genetic admixtures into the crossing program, okay. Otherwise, if you are going to cross between, if you are supposed to cross between two parents, two desired parents, okay, and if there is a foreign pollen from undesired parents, okay, so maybe wild, okay, which is more more viable pollen, which has more viable pollen, so it will pollinate instead of your desired parent, okay. So in this case, so it is quite necessary to bag the emasculated flower and pollinated flower, okay. Okay, pollination program, okay. Right after emasculation, in the next morning, emasculation is usually done during evening, okay. 4 to 6 usually it is recommended, okay. And after, usually after sunset, and right after that in the next morning, early morning, pollination is done, okay. So pollination is is pollination can be done with the help of a brush, okay, which is what quite commonly used method. If the amount of pollen is less, if the amount of pollen produced by the pollinators, or you can say male flower, okay, is less, so we are going to collect the pollens with the use of brush and smear it in the stigmatic surface of the emasculated flower. Okay, so what we are going to do is we are going to grow two different plots, one with pollinators, or you can say the desired male parent, and the second one being your emasculated flower. Okay, the parents you want to act as a female parent. Okay, so in this, the flowers from the pollinator plot is going to be collected and it can be pollinated into the desired field plot where the pollens are supposed to the pollination are supposed to happen. Okay, and the pollination can also be done by directly bringing the flower from the pollinator plot or male parent and then smear it on the emasculated flower. Okay, this can also be happen, and it can also be done with the help of earbuds, okay, or any other mechanisms, DIY, whichever it is found effective. And next is tagging, which is most important thing, okay, because right after you do emasculation or pollination, if you don't keep the record, okay, it will be lost. Okay, there will be confusions, there will be multiple flower buds and there will be lots of confusions. Okay, so that is why it is important that you need to have a tag, okay, where the details of the date of emasculation, date of pollination, date of and then parentage, okay, and the number of flowers emasculated has to be written into the tag. Coming to the emasculation and pollination techniques in tomato. Okay, first of all, when we look into the emasculation process, in tomatoes we are going to select those flower buds which are on the right stage. Okay, not too mature, not too immature. Okay, so this means that the flower which hasn't started its anthesis, okay, that means the flower which hasn't opened yet. So this is already open one where the pollen grains are already activated and the cell fertilization has already taken place in this. So the seed or the seed which is produced by, or the plant which is produced by this flower will be self pollinated plant. Okay, and in this, since there is no pollination taken place, so we can remove the male flowers from this. Okay, and then before anthesis starts, and then we can pollinate the stigma with the foreign pollen, a desired foreign pollen. Okay, and you can see also here the flower buds which are immature, we have to avoid this. Okay, because there is of no use if you pollinate this one because they are not in the right stage. Okay, and the flower color, to select the type of flower color, it should be the anthers and corolla are beginning to change from light to dark yellow. Okay, so it will slowly convert from pale yellow to dark yellow. Okay, and this so-called emasculation technique has to be operated well before anthesis. Okay, and it comes with experience, or you can say skill, when and how to do emasculation and pollination program. So it comes with experience. So after doing few trials, you will be able to identify those flower buds which are in the right stage for emasculation and pollination. Okay, so next thing is tomato pollination. Right after pollination we are going to do right after emasculation we are going to do pollination. Pollen, which is the source of male the male flowers which are the source of pollen grains, okay, are to be collected, or are to be the pollens are going to be collected from the male lines. Okay, and it may be with the help of forceps, or it can be with the help of brush or any other means. Tweezer, it may with the help of earbuds. Okay, so the collected pollens are going to be smeared into the stigmatic surface, and it is to be uh, it is to fully cover the stigmatic surface so that the pollen germination is starts significantly right after the pollination program. Okay, so after the pollination has done, so it has to be tagged with a red thread, tied with a red thread to indicate that these emasculated flowers are pollinated. Okay, so these few things you have to take care into account, because in order to avoid confusions, okay, red thread can be used to indicate pollinated flowers, and few things also can be taken care, like removal of forceps which are going to be used for pollination and emasculation have to be sterilized by alcohol and methylated spirit, because any kind of infection may happen, fungal or bacterial infections may happen, which may damage the young flower buds where there is no protections. Okay, next thing is brinjal. Next crop is brinjal. We are going to discuss here emasculation. And the flower generally emerges right after 40 to 45 days of transplanting. Okay, and anthesis occurs during August and September at 6 to 8 a.m. Okay, which is quite early compared to December, January during winter, because of low sunlight, low light intensity. The anthesis or flower opening occurs a little bit late, which is 9:30 to 11:15 a.m. So right after that enters usually dehis. Okay, right after anthesis. Okay, anthesis is what? Flower opening. Okay, enter dehiscence is another thing, which is uh, dispersal of the pollen grain. Okay, so right after anthesis, right after opening of the flower, the dispersal of pollen happens after 15 to 20 minutes. Okay, for emasculation, a healthy long medium style well developed bud from central portion of the plant is selected. Okay, and the bud is opened gently with the help of fine pointed forceps or with the help of a pointed needle. Okay, and one or 2 days before the opening of the flower bud, and all 5 anthers are carefully removed. Okay, so since the flower in case of brinjal is a little bit large compared to other crop plants, so there is no issue of uh, using micro you can say lens, or because sometimes lens is used as a mechanism to identify the androecium. Okay, in case of small flowers, so this is of no issue in this, so you can identify easily the five anthers which comes along with the stigma. Okay, so you can directly remove this along with the corolla. Okay, because now we are going to bag it, so there will be a protection. Corolla's role is going to be replaced with a butter paper bag. Okay, right after emasculation. Okay, right after emasculation we are going to do pollination in the next early morning. Okay, so this is a usual uh, process, okay, of emasculation and pollination, where emasculation is usually done in the evening, okay, after sunset, and pollination is done in the next morning. Why emasculation is done in the evening? Because if you emasculate in the morning itself, because you have to wait after emasculation, you have to wait for 1 day. If you do so, in the all the uh, what you can say, your moisture content and the flower may dehisce, okay, nutrient may dehisce, okay, because of exposure to sunlight. Okay, and in the next morning, if and if you do it in the evening, so this kind of problem is not there. Okay, because there is no light. Okay, so in the next morning you can directly uh, pollinate with the help of forceps or tweezers, or you can say brush. Okay, so pollination usually remains viable for a day during summer. Okay, so it has to be done during the emasculated flower has to be pollinated the next day with pollen. Okay, 2 to 3 days in winter under field conditions. Okay, right after anthesis. Okay, the pollen viability is stays for 2 to 3 days. Okay, and for pollination, freshly dehisced anthers are picked up and are slit vertically with fine needles to get sufficient pollens at the tip of the needle. Okay, so this is also a case of in case of uh, male uh, male plants. So we have to collect uh, the matured flower. Okay, unlike that of female plant. Okay, we have to collect flowers where there is yellow colored pollen grains all over the anthers, and then we have to collect these, and either we can directly emasculate by using flower to flower, or you can collect the pollen grains and smear it with the help of a brush. Okay, and right after that, as usual, it has to be labeled with a tag and covered with a small pollination bag. Okay, so coming to the end of the slides, if you have any questions, you are open to